Welcome, everyone. Thank you for being here today. I'm Bill Sanders, the William D. and Nancy W. Strecker Dean of the College of Engineering. In 2020, the College of Engineering kicked off a new webinar series entitled Faculty Insights, a 20-minute briefing, which we're holding 5 times per year. In this virtual series, faculty from across the College of Engineering share insights into their research, its impact, and a perspective for the future. Following each 20-minute presentation, attendees will have an opportunity to direct questions and comments to the speaker. Our speaker today is Professor Kathryn Whitehead, Professor of Chemical Engineering. The COVID-19 mRNA vaccines upended the course of the pandemic and the world's approach to vaccination. At the heart of those vaccines are nanoparticles that package up the fragile RNA and deliver it safely into the body's cells. Today's presentation, entitled Nanoparticles for RNA Therapeutics, Enabling a Medical Revolution, will focus on Professor Whitehead's lab's role in engineering lipid nanoparticles for next-generation RNA therapeutics with the potential to treat diabetes, cancer, cystic fibrosis, neurological disorders, and more. Katie joined the Department of Chemical Engineering at CMU as an assistant professor in 2012. She earned her bachelor's degree in chemical engineering from the University of Delaware and her PhD in chemical engineering from the University of California, Santa Barbara. As a graduate student, Katie developed systems for the oral delivery of macromolecules. From 2008 to 2012, Katie undertook postdoctoral training at the Koch Institute for Integrative Cancer Research at MIT, where she developed biomaterials and methodologies for the advancement of RNA interface therapeutics. The research interests of the Whitehead Lab lie at the interface of chemical engineering, molecular biology, and medicine. Her group's ultimate goal is to engineer safe and effective drug delivery systems capable of achieving therapeutic outcomes in biological models and ultimately in humans. As a first step, Katie's lab is interested in developing a fundamental understanding of the relationship between delivery barrier biology and drug transport. To accomplish this, her group is employing RNA interface, a biological phenomenon that induces gene silencing in the presence of siRNA. Through identification of the cellular components essential to the drug transport process, Katie's group is able to design drug delivery systems using modern chemical techniques to overcome or cooperate with those components. Before I turn things over to Katie, please know that you can submit any questions you have via the question and answer feature at the bottom of your screen. You may do this at any point in the program. Be sure to include your name with your question. Tiffany Sudar, Assistant Director of Development for the College of Engineering, will be monitoring the question and answer session with Katie at the conclusion of this presentation. Katie, we're very much looking forward to your presentation. Thank you for being here with us today.
Thanks very much, Bill. It's a pleasure to be with you all here today and to share some work that my lab has been doing in the area of RNA therapeutics. So, as Bill mentioned, I've been at Carnegie Mellon now for a little over 10 years. I moved to Pittsburgh at that time. I'm a Pennsylvania native. I grew up in Allentown over on the other side of the state. And it's just been a wonderful place to begin my career as an academic. It's been a wonderful, supportive environment with great people. So just a little bit about myself. So I'm an engineer and professor, and during my time here, I started a family. So these are what I refer to as my startup company. And people ask me what my hobbies are. Many of them have disappeared, but my main hobby right now is trying to raise nice children, as well as continue to grow tomatoes, which are my favorite. We live in Squirrel Hill, so I know many of you are familiar with the area. So that's where we are, nearby. And I love it there. I'm very close to 5 Points Bakery, if anybody's had the treat of going there. So my lab, as Bill mentioned, focuses on drug delivery. And drug delivery as a science is how we package up drugs to take them to places that they need to go inside of the body. So there are a lot of medications that you might take. You can think of something like Advil, where you take it orally. It goes literally everywhere in your body. So if you have a headache, it'll help your headache. If you have an ache in your leg, it'll help the ache in your leg. And it doesn't really matter because it's very safe and it goes everywhere. But there are a lot of medications that you might want to take where you really don't want them to go everywhere, or there's some kind of problem in getting them to where they need to go. And so that's where drug delivery scientists come in to try to engineer solutions that will make the most effective and safe drug possible. So we have a couple of different thrusts. One is in the area of oral protein delivery. So we try to develop methods to take protein medications like insulin, and instead of having those injected regularly, you can instead take an oral formulation, which is much more desirable. We study applications in women and infants' health. When I became pregnant at this point 6 or 7 years ago, it was never more apparent to me the, shall I say, pathetic state of maternal medicine that's out there. And it really has motivated me to find some of our drug delivery systems and see how we can apply them to not just create new medications for women and their infants, but also just to learn anything at all, because there's really so much to do in this space. And I'm just so happy to have entered this field. And then finally, what I'll be talking about today is our work in RNA therapies, and specifically in creating the nanoparticles that are used to take these RNA drugs to where they need to go inside the body. So when I began my lab 10 years ago, RNA therapies were still considered very much a niche technology. I've been working in the field for about 15 years, and the reason why I got into it is because when I learned about this concept of personalized medicine and gene-based medicine, 15, 20 years ago, I just thought this is going to be the medicine of the future. This is going to be all of the solutions that we are going to need to treat disease. And I wasn't sure how long it would take to realize these types of therapies, but I knew that I very much wanted to be involved because I believed in it very much. And so over the course of the 15 years that I've been in this field, we've made an awful lot of progress. And so in the beginning, I actually had a lot of trouble finding funding for this work because so many other scientists really believed that it would never amount to anything. So it's really been gratifying to see how much it has grown over the years. So this is the first RNA therapeutic that was FDA approved. That happened in 2018, and this was just like a really beautiful moment for all of us in this field. Incidentally, this drug contains lipid nanoparticles that are very similar to those that I'll be speaking about today. And then, of course, we had the horrible occasion of the pandemic to bring forth these mRNA vaccines that have now saved so many people around the globe. And whereas you could argue this first FDA approved drug, this is for a rare disease indication, it wasn't necessarily clear that these systems could be deployed in a large number of people. But given the pandemic, we've now seen safety and efficacy in literally hundreds of millions of people worldwide. And so the floodgates are now going to open in terms of really taking RNA therapies more seriously. And so I think, we already have hundreds of clinical trials that are now happening with different types of follow-on medication. And I think it's only going to go from there. So, excuse me, why is RNA so exciting? as a potential treatment. It all comes down to the molecular or the central dogma, of molecular biology, which is here. It tells us that DNA, which is our permanent genetic blueprint, this is transcribed into a temporary copy called messenger RNA, which is then translated into protein. Now, proteins are the doers in our body. They're responsible for nearly all function that is present inside of us. And disease is caused by problems with our proteins. Either we have too few of a protein, we have too much of a protein, perhaps we have a mutated protein that's not working properly. All of these issues are caused by proteins. And so if we want to get to the root of disease, we need to be able to fix that protein expression. There are a couple of different ways that you can do it at the protein level, at the messenger RNA level, or all the way up at the DNA level. And there are pros and cons to each. We believe in my lab that at the messenger RNA level, we have a lot of flexibility. We're introducing a molecule that exists temporarily, but a single messenger RNA molecule can create many, many molecules of proteins. And so we can really have nice effect this way. And there are a couple of different ways we can control protein expression. One is with a short piece of RNA called short interference RNA. This can interact with messenger RNA to turn off or turn down protein expression. And then we can also ask our bodies to make more of a particular protein by delivering messenger RNA for a protein perhaps that we're already making, that we want more of. Or as in the vaccines, what's really cool is we can deliver messenger RNA that encodes for proteins that our bodies have never made before. We can provide completely new instructions to our bodies and ask them to make something new. And that's what we have in vaccines, where we're trying to make proteins that are antigens that reflect little portions of viruses, for example. So because of this ability to affect protein expression, mRNA and other RNA drugs are really well poised to completely revolutionize medicine as we know it. And there are boundless applications, and I'll just name a few here. So one is protein replacement therapy. This is the most straightforward application in which you have a disease, perhaps hemophilia, where somebody is missing a protein. So in the case of hemophilia, you're missing a blood clotting factor in your blood that helps you clot. And so you can deliver messenger RNA into the body, and you can create this protein, and you can treat the disease. The most clinically advanced, of course, are vaccines. We can also, it's similar to vaccination, you can also use mRNA delivery for cancer immunotherapy, in which we can train the immune cells of a person with cancer to go after and attack the cancer cells, the very specific personalized cancer cells that are in their bodies. We can use messenger RNA to perform regenerative medicine. So this is if we want to take stem cells and we want to create a new liver, or we want to create new bone muscle, we can help in that process. And all of these involve making more of a protein, but we can also use messenger RNA to engage in what are called gene editing strategies, for those of you who have heard of that. And in this way, we can also turn off gene expression. And so it's a very powerful technique where we can turn on or turn off different proteins and really treat just about any disease that's out there. So the problem with RNA is that it is a very fragile molecule that the body readily degrades if you introduce it into our bodies. It's also very large, so it would get cleared through your kidneys pretty rapidly. It's not going to go into the cells where it's needed, and it's very negatively charged. So every base on the RNA, it has a negative charge. And because of that negative charge, it's actually repelled by the cells in our body, because the membranes of our cells are also negatively charged. So these RNA molecules really need help when it comes to getting to the right place inside the body. And I just want to show you a couple of the barriers that these delivery systems need to overcome, because I think it's amazing that we found anything at all, quite honestly. So if we put our RNAs inside these lipid nanoparticles, which are the particles I work with, we inject them, say, into the bloodstream or perhaps into the muscle for a vaccine. And the first thing that needs to happen is they need to avoid being gobbled up by the immune cells in our bodies. We don't want them to get cleared right away before they've gone someplace. They need to exit the bloodstream into some tissue called the extracellular matrix. So we need to get to that cell type that we're really interested in. The cell then needs to uptake that nanoparticle to bring it into the cell, into a compartment called an endosome. And that endosome is pretty tough. It's like a garbage can. It would normally destroy our nanoparticle. So one of the most difficult parts of this process is to design the nanoparticle chemistry so that the nanoparticle can kind of bust out of this trash can into the main part of the cell, where it then can ask the cell to make the protein that we're interested in. So we're asking a lot of our nanoparticles. And when I got started in this field, our goal this whole time has been to develop potent and degradable RNA delivery systems. Potent because we want to, get as much drug as we can into the system, and degradable because we want to have the opportunity to do repeat therapies. And if you're going to be delivering a drug again and again, of course, it's important for it to not build up in your system over time. So over the years, to accomplish this goal, we've screened thousands and thousands of lipid nanoparticles from lipids that we have synthesized in my lab. We've used combinatorial chemistry, meaning that we take different components and then we combine them together in many different conformations to make a wide library of materials. And the reason we do this is because it's not particularly well understood how the chemistry of a lipid then affects how well it's going to deliver the RNA. And so we try to figure out structure-function relationships and how we can improve upon previous generations of materials. So how do these lipids work in encapsulating the RNA into nanoparticles? So here's one of my favorite lipids that we've developed in my lab. We call it a magic lipid, and it will take on a positive charge. And this positive charge is important for a number of different processes during delivery, the first of which is forming the nanoparticles in the first place. So again, the RNA is very negatively charged. And so you have this really nice electrostatic interaction that helps the lipids condense the RNA into a nanoparticle. And this is typically accomplished for the vaccines as well, using microfluidic systems in which we rapidly combine an aqueous solution that contains RNA and an organic solution that contains different types of lipids. And when you rapidly mix them together, you have nanoprecipitation that occurs, and you have these nice nanoparticles that are going to come out the other side, which tend to be about 80 to 100 nanometers in diameter. And each one of these lipid nanoparticles contains a number of ingredients. Typically, they contain 4 lipid ingredients in addition to the RNA. And each one of these lipids plays a very important role in the delivery process. I don't have time to go into all of them, but I can mention that this ionizable lipid, this is usually where IP exists. So my lab has its own IP. We use our own ionizable lipids. The Pfizer-BioNTech vaccine uses a proprietary lipid. The Moderna vaccine uses a different proprietary lipid. And then some of these are naturally occurring molecules. Cholesterol, when it's in a nanoparticle, it's definitely not bad. Each one of our cells, the membrane surrounding our cells, it has a ton of cholesterol in it. That's where cholesterol is good to have because it provides structure and stability. We would all actually be piles of goo if it were not for the cholesterol that are in our cells. And so our nanoparticles need them for a similar reason. So the challenge that we have in terms of finding a nanoparticle that works well is then can we deliver it to specific sites inside the body? This is not a system where you want to put a nanoparticle in there and have it go everywhere necessarily. And it can actually be very difficult to deliver to different types of organs and tissues. So that's been one of the goals of my lab. And if we look at the products that are currently out there and FDA approved in terms of RNA therapies, there are really just a couple of targets that are hit again and again. So almost everything in clinical trials in terms of protein replacement is for the liver. And this is because when you inject nanoparticles into the bloodstream, the liver is going to be the first organ that tries to filter these nanoparticles out. And so although I don't want to say it's easy to deliver to the liver, it is easiest. Okay, so, that's where a lot of the commercial successes have been so far. And then in terms of vaccines, we've also seen some very nice delivery to the immune cells inside the muscle for vaccination purposes. However, as you can imagine, most diseases are all over the body. They are not in the liver necessarily or in the immune cells within the muscle. And so we have an awful lot of work to do in terms of trying to figure out how to deliver our RNA everywhere else. Okay, a lot of diseases occur in these other parts of the body. And so as chemical engineers, we look at these lipid nanoparticles as a treasure trove in terms of formulation space. We can change just about everything you can think of with this nanoparticle. We can change the identity of each one of these ingredients. We can change the molar ratios of the ingredients. We can add or take away ingredients. We can change the type of RNA that we use. We can put targeting moieties like antibodies on the outside if we want to. And I think there's a rhyme and reason for all of it. How is it that the chemistry of these systems are affecting where they're going inside the body and what they're doing? And so that's really what we want to figure out in order to unlock delivery to all of these interesting places. So I don't have the time to go into it too much in depth, but I want to share with you some of the exciting work that the people in my lab have been doing to take this RNA to new places. So earlier this year, we had the first publication ever in terms of delivering RNA to the pancreas. And I'm just really delighted for the postdoc in my lab, Jillian Mellon, who had done this work. She was really motivated to try to find a delivery system to go to islet cells inside the pancreas. So for all the work that we do in my lab, we begin with mouse models. It's very important to use a living system where there's circulation and we can figure out where exactly these particles are going in terms of organ distribution. So here I'm showing you the successful experiment in which mice have received our RNA treatment. And these are slices of the pancreas that have been taken. So this is an untreated mouse. And here are structures inside the pancreas that are called islets. And the islets contain a couple of specialized cell types, one of which are beta cells. Beta cells are super important because they're the cells that make insulin, and they're the cells that are attacked by your own immune system during diabetes. And so they're really destroyed. And the ability to deliver something to islet cells would be absolutely huge because it would open up potential therapy for diabetes. And there's also a specific type of pancreatic cancer that occurs within these islets. And so what Jillian was able to do is to change the chemistry and the way that we were injecting this nanoparticle to create protein expression in the pancreas. So here she delivered messenger RNA that encodes for a protein that's showing up as brown right here. And you can see all of this brown signal inside of our islets. So really delighted with her ability to do this, and we're moving forward with looking at potential applications. I have another talented postdoc named Sai Erneni. He was a PhD student in biomedical engineering, and I was really fortunate to have him join my lab about 1.5 years ago. So he's developed a system for delivery to the brain. This is accomplished using an intrathecal injection, so very similar to how women receive epidurals during childbirth, and they're used clinically in a number of scenarios. So that allows us to go directly to the brain, and then the quality of the delivery system is important, and he's been able to deliver to many of the different cell types inside the brain. So here I just want to show you some pictures. Here is an untreated mouse brain. You can see we don't have any protein expression here. This is firefly luciferase, so we can see the brightness kind of like in a firefly that we can pick up using an imaging system. This is a fluorescent protein that these brains are also producing. That's red. And what's really cool here is that he can deliver not just this one mRNA, but he's actually delivering 2 mRNAs at the same time. So you can imagine that some diseases, there's going to be more than one problematic gene or protein involved. And so it's exciting that we have delivery systems that are potent enough to deliver more than one at a time. And also importantly in this system, but also in our other organs that we're delivering to, if we look at all the other organs in the body, we don't see any signal there. And so that shows us that we have this really nice organ and cell-specific delivery system where we don't have to worry about off-targeting or potential side effects in these other sites. The same postdoc, Sai Yerneni, has also developed a delivery system for the lungs. And in this case, we're looking at intranasal delivery. This can be used potentially for alternative vaccinations, so instead of intramuscular injections, and also potential therapy for cystic fibrosis and different types of lung cancer. This is one of the my favorite images ever produced in my lab. This is a picture of mouse lung to which Sai had delivered messenger RNA. And the protein that the cells made is in green. So wherever you see green, that's where delivery happened. Each one of the cells here, or I should say, this is an airway. So the black part here is an airway through which the air would typically come, where the nanoparticles probably came down. And then these are the surface airway cells, and they're all beautifully expressing this protein here. And it's hard to tell if you don't know what you're looking at, but there are some cells here kind of in the back, hiding in the back. These are actually lung stem cells. And so if you have issues like cystic fibrosis, which are permanent genetic disorders, by delivering to these stem cells, which Sai has done, you open up the ability to permanently correct some of this dysfunction that's occurring inside the lungs. And then finally, back to my interest in pregnancy treatments, we're interested in seeing how lipid nanoparticles can be used for delivery to the placenta. So the placenta, there can be all sorts of different disorders of the placenta during pregnancy, the most common of which and the most deadly of which is pre-eclampsia, which is the condition in which a pregnant person's blood pressure is sufficiently elevated to the point where they have to prematurely deliver the infant because it's really the only treatment that's available. So it would be great if we had some other alternative than inducing childbirth. So here, my PhD student, who's now graduated in a postdoc at MIT, he developed a system that can deliver quite nicely throughout the placenta. So this is an untreated mouse's placenta, and all the blue, these are the nuclei inside the placental cells. And then he was delivering RNA encoding a red protein. And so you can see throughout these different sections within the placenta, we have some nice delivery. And very importantly, so here you're seeing on the y-axis, this is the amount of delivery that we have. Very importantly, we have delivery occurring in the placenta, and we do not have any delivery occurring in the fetus. And so we want to keep the fetus completely unexposed to the drug and make sure it's a safe system. So this is a snapshot of some of what the lab has been able to do in terms of changing chemistry in order to meet these needs. And so in thinking about where we're going to go next, First, we would like to identify, better understand the mechanisms of action of the delivery systems that we've already made. I have a little schematic here showing a type of immune cell called a macrophage. And we believe that macrophages in these different tissue and organ types are crucial to what we're seeing in terms of efficacy. And this is really interesting because we normally think of the immune system as our enemies in terms of delivering drugs, because they're very good at clearing anything that's foreign. But there's actually something very special we think that is happening, and we want to better understand it, because if we better understand it, then we can improve the quality of our delivery systems and hopefully be able to go to even more organ targets. Which is our next goal, to go to places such as the bone marrow, where there are a ton of hematopoietic stem cells, to be able to go to cardiac and other types of muscle tissue to treat diseases such as muscular dystrophy. And there are a host of different ways that we could go about this in terms of working with more lipid nanoparticles, changing the type of chemistry to something like a polymer, taking completely new approaches. We're also interested in creating more patient-friendly administration routes. So a lot of what's already out there is injection-based, and that's not always ideal for reasons that I'm sure many of you can appreciate. So developing oral delivery systems, or for example, intranasal sprays that are going to be less invasive, is something of great interest to us. And then, of course, finally, our ultimate goal here is to treat disease. And so now we're armed with these delivery systems that are capable of going to these unique organ and cell targets. And so we're now partnering with clinicians, mostly at the University of Pittsburgh, but also in other parts of the country. I'm just starting a company right now with a physician out at UCSF, for example, that's going to focus on RNA therapies for neurodegenerative conditions. And we also work with a couple of clinicians here. We work with a pancreatic surgeon over at Children's Hospital on type 1 diabetes treatments. And we also work at the McGee Women's Research Institute. We have a number of people who are interested in placental biology and the development of medications for pregnant people. So thank you so much for being here today. I would be remiss if I did not acknowledge the absolutely wonderful people that I've worked with here at Carnegie Mellon, my PhD students and postdocs throughout the years who have conducted this work. It's truly a very special place to do research. I'd also like to thank my funders that have directly funded some of our RNA research over the years, and a special shout out to John Sachs and his partner, Myrna Marshall. John is an alumnus of the college, and they've been generous in their support of some of our work over the years. I will also point you, if you're interested in learning more about the nanoparticle ingredients and where I think the field could be going. I had the privilege of giving a TED Talk a couple of years ago on what they call fat balls, which are my lipid nanoparticles. And I'm also on social media if any of you are there and want to connect. So thank you again for joining us here today, and it's my pleasure to take any questions that you have.
Thank you, Katie. We very much appreciate all of these great insights that you've shared today. And now we'll open it up to the audience for any questions you may have. Just a reminder, please use the Q&A feature at the bottom of your screen so that we can queue up your questions and get as many as possible answered here today. And there will be a follow-up email, which I'll tell you a little bit about later, that will also cover some of these things. So I'm just going to kick things off right away, Katie. First, we have a lot of people thanking you so much for your drive to optimize these nanoparticle and RNA therapies. I personally appreciate that. Hopefully there will be some new ways of drug delivery that doesn't include needles. So thank you for that. But our first question comes to us about about how close is your work getting to viable breakthroughs with diseases such as age-related macular degeneration, Alzheimer's, Parkinson's, sort of those diseases. So if you can talk a little bit about that.
Sure. So age-related macular degeneration is a disease of the eye. And so right now my lab isn't focusing on diseases of the eye or delivery to the eye, but I certainly have colleagues in other parts of the country who have developed nice systems for delivery to the eye and who are pursuing treatments for those types of diseases. As far as the brain delivery work, we just discovered the delivery system about 9 months ago. And so in terms of developing therapies, usually, even with the remarkable partner that I have, I think it would at least be 3 to 4 years before we would have something in a person. And that would be for diseases that, where there's a more established genetic cause. So one of the first that we're going to look at is ALS. There is a more established genetic cause there. The challenge with Parkinson's and Alzheimer's is that the genes that are responsible are a little bit less clear. So that's why it's important for us to also partner with the biologists, because as they learn, then we can really deploy these solutions.
Great. And Katie, you do a lot of sort of work and collaboration, particularly with our biomedical engineering department, but also all of the departments in some way or another around this. So can you talk a little bit more about that collaboration and sort of how those, non-existent barriers to entry are helpful in your work here?
Sure. So collaboration is absolutely a critical component of what we do. It's a very interdisciplinary lab, and I have some of the more traditional chemical engineers sometimes asking me how this is chemical engineering. I think it's a little bit of everything. So we use molecular biology techniques. Believe it or not, I do the most organic synthesis of all the chemical engineers in my department. So we do a fair amount of chemistry. I do collaborate a bit with the people over in chemistry because we're always trying to make new chemistries, and I'm not actually a chemist by training, so it's very helpful to work with them. Over in MCS, there's also, it's housed in MCS, is the Center for Nucleic Acid Science and Technology. And that's a group of faculty that are very focused on nucleic acids, the biology of them, but also then in developing therapies and treatments. And so it's been a huge help to me over the years to be able to tap into those resources. And then within biomedical engineering, we have a lot of imaging strength in biomedical engineering, really nice microscopes, and working with people to really understand at the cellular level what's happening with our particles. And then I'll also mention I'm working with Phil LeDuc over in mechanical engineering. We're developing some microfluidic systems, so it's nice to work with microfluidic experts as well. So really, it's a very multidisciplinary field. I say as a chemical engineer, I belong in this space because it's important to, I think, come at some of these engineering problems with that engineering angle. I'm here to fix a problem. And as a chemical engineer, I believe that it's the chemistry of these systems that will allow us to fix it.
Thank you. I'm just going to keep going with these because we've already had a lot come in, and I want to make sure we can get through as many as possible. So shifting a little, what regulates siRNA, and what is the role of epigenetics and PTM, post-translational modifications? How are these a risk or opportunity for gene therapy?
Well, I don't think so. siRNA, short interfering RNA, this is not something that is natively produced inside the cell. So this is something that we engineer and create outside the cell and then deliver inside of it. It is not involved in epigenetics. Epigenetics occur at the DNA level. So, that's that's not so relevant to epigenetics. What's interesting is we are trying to take approaches to look at epigenetic regulation. There are some new and exciting CRISPR and Cas9 based tools that will allow us to, for those of you who don't know, DNA inside the nuclease, there's something called epigenetics. So genetics is like what the DNA code actually says, and epigenetics are little tags that are put on and off, taken on and off of our genes to basically say, hey, make this gene, don't make this gene. And so you can actually fool around with the tags that are on the genome to try to control protein expression that way. So that's a very new area. And yes, we're trying to make some headway there.
The next question is, for background and context, what percentage of traditional medicine to treat disease that is not RNA interventions directly addresses the protein that is causing the problem?
Well, it's hard for me to give an exact percentage. So a lot of medications are designed to treat symptoms and not actually fix the problem. So there are some medications, for example, cancer therapies, anything with the term inhibitor on it. An inhibitor is always going to be changing something about protein expression. You're trying to inhibit the function of a particular gene or protein. There are other antibody-based drugs. So antibody-based drugs are generally working to change the expression of proteins in a cell. So there are certainly some of each. I couldn't really give you an exact percentage.
Another question that might be related to this a little bit is, how does your technology differ from C-dot nanotherapy coming out of Weill Cornell?
So carbon dot nanotechnology is just a completely different ballgame. It's just made of completely different materials. The commonality is that there are nanoparticles involved. C-dots are generally going to be smaller than the particles that I'm working with. And so, they could be used for different types of applications.
And then, are there cases where you might want the drug to persist in the body rather than degrade?
Yes. So different types of disease, you may want to upregulate or turn on protein expression for a short period of time. Perhaps you want to turn it on for longer periods of time. Sometimes, in the reverse, maybe you want to turn something off for a little bit or more permanently. And so there are different combinations of types of RNAs or other nucleic acid drugs that we can use to try to change the exact nature of the regulation that's going to occur. It also depends in part on the exact protein that you want to regulate. Some of them, for example, have longer half-lives than others. Messenger RNA, if you want to permanently turn something on, it's not the best choice in that case, because the messenger RNA will create protein for some period of time. Depending on the protein, it might be a day, it might be a month, but it's definitely not going to last forever. There are some diseases, though, where you only need it around for a little while, and then you've fixed the issue. And so it really depends on the nature of the condition.
Thank you. And then this is sort of a three-part question. So, but I think it all goes together. So I'll ask you all at once. Is ribosomal and transfer RNA involved in the process in which you work? Are the RNA material made from organic or inorganic substances? And finally, does diet play any role in the effectiveness in these RNA treatments?
So rRNA and tRNA are not, I would say, directly related. So tRNA, for example, transfer RNA is an RNA kind of helper in our cells, and it helps to make the proteins and line up the amino acids in the right way that are going to generate the proteins. So all of these RNAs are important for protein regulation inside the cell. However, the typical delivery of messenger RNA is not going to, mess around with any kind of tRNA functionality. In terms of RNA, so RNA is made entirely of organic components. It's all carbon, hydrogen, nitrogen, oxygen, and phosphate. So that's all organic. And then does diet play a role? That's an excellent question, and I don't know the answer to that. There are some people who are looking at the influence of fat percentages on delivery. I don't know how much it would really affect the efficacy, but the question is, depending on what kind of fat content a person has in their body, does that change where the nanoparticles go a little bit? Do they like fat? Do they not like fat? There's still a lot to discover there.
We're going to do a few more here. One is, could you say more about the role of macrophages in your systems, and in general, how you address the risk of immunogenesis?
So macrophages are only one component of the complex mess that is the immune system, and we're learning more about it all the time. So in terms of immune responses to these materials, it's not even so much the role of the macrophages that can kick that off. So it has more to do with, there are some innate immune responses that occur. There's a receptor in your body called toll-like receptor 4 that is involved in recognition of foreign lipid molecules. And so that can sometimes kick off some inflammatory processes. And there can also be adaptive immune responses in which these particles are then cleared from the bloodstream if there are antibodies that can actually form against particles that will prevent them from being used again. Nobody advances those to the clinic, right? You find nanoparticles that can create these sorts of responses, but those are never the ones that you move forward with. And again, it's something about their chemistry that provokes these unwanted immune responses. What I mentioned a bit about the macrophages, what I can say is that it looks like macrophages are playing a role in how these nanoparticles are working, in that they are uptaking the particles. They take in the mRNA, and we are not the first people to discover what's called horizontal gene transfer. It's a natural process in which cells talk to each other, and they can send messenger RNA and other material from one cell to another. And it seems as if the macrophages that take up our particles are then sending some of it to other cells. And that's how it's actually going into the other cells, as opposed to our nanoparticles getting into those cells directly. So they're kind of like this in between. And, there are a lot of questions as to exactly how this is happening.
And then what are, or why are the lipids proprietary?
Well, they're not naturally occurring molecules. And so people have had to make them in a lab and try to figure out what is the, best kind of structure to make these with. And so whenever you make something that's new, that nobody has thought of before, you have the potential for intellectual property. And that's where a lot of the intellectual property is in the lipid nanoparticle space in general. And that's why, different companies might be able to create different products because of that. They all use cholesterol, for example. They all use the same other lipids that are in the particles. And so they don't have any intellectual property in that space. It's really these ionizable lipids.
And then I think we may have touched on this a little bit, but if you want to dig any further, do neurodegenerative diseases include Alzheimer's? And is there anything promising with that disease in particular?
So yes, they do involve Alzheimer's. And, as I mentioned, I think we need to understand a little bit more about the biology before we really have a path forward. But, depending on how some of the earlier work goes with some of our initial efforts in ALS and dementia, there might then be an opportunity to move into Alzheimer's.
Okay, great. Let's see. Make sure I cover everything that seems like we haven't covered yet. And on the slides where you were sort of showing the mouse and the things that were lighting up in the different body parts, for that, is that showing protein expression in those images, or is that just the delivery system itself?
That is the protein expression that results from the delivery.
Okay, great. And then let's see. Are your lipid nanoparticle delivery systems inclusive of CRISPR and Cas9 treatments?
They can be. So most of what we work with don't incorporate CRISPR Cas9. However, mRNA is, in my opinion, the best way of going about CRISPR Cas9 and gene editing therapies. And the reason is because what you do is you deliver messenger RNA that encodes for Cas9. So Cas9 is a nuclease that can go into the cell. We also deliver what's called a guide RNA, and the guide RNA will go with this nuclease into the DNA, and it'll figure out where exactly to sit down and make a cut. Okay, so that nuclease, which makes the cut, it's only around for a short period of time because the messenger RNA got in there. It made this thing can go into the DNA, make the cut. Whereas a lot of people, they deliver DNA that encodes for this Cas9 cutter. And when you deliver DNA, this thing is going to be cooking around for a lot longer. And I don't know about you, but I don't want something that's cutting my genes to be around any longer than it needs to be. So I think messenger RNA is a pretty exciting option for gene editing.
And so again, if anybody has questions, just please remember to put them in the Q&A feature at the bottom of your screen so we can answer as many as we can in the next few minutes here. But I'm going to switch over to two questions that sort of are both about the COVID vaccines. And so just to get an understanding, in the case of the COVID vaccines, was the point simply to employ muscle cells to generate protein to induce the immune response? And then in other cases of organ delivery, are you focused on getting mRNA to specific types of cells to modify their DNA and or induce those cells to produce a given protein?
So with the vaccines, you're delivering messenger RNA into the muscle. What is transfected, what actually takes up the messenger RNA? There are some muscle cells, but the main target are the immune cells that reside within the muscle. They're the ones that have the most active immune response. And so in that case, the messenger RNA encodes for part of the spike protein. The spike protein is produced, and then you can kick off this nice immune cell response. I was reading the rest of the question. So the rest of the question was, what was it?
Oh, let's see. Let me just get you the rest of the question.
Sorry, let's see. It's answered call.
Okay. In other cases, organ delivery, you're focused on getting RNA to specific cell types and DNA.
Yes.
And to induce those cells to produce a given protein.
Yes. So we won't be modifying their DNA. Generally, when we use messenger RNA, unless it's for gene editing, which is, not for most of what I've been talking about, we don't want to modify the DNA. We want to go in and we want to ask those organ cell types to make a particular protein.
And then, so what is the nanoparticle size and how much does that matter in sort of these delivery systems?
Sure. Yeah. The nanoparticles tend to be about 80 to 100 nanometers in diameter. I get this question a lot it's very species dependent. So in mice, the nanoparticle size doesn't matter a whole lot, but all those barriers that I told you about in the body, you can think of these barriers actually have different sizes in different physiologies. So there are holes in the bloodstream that go through the liver. They're called fenestrae. They're basically the gaps through which these particles can go through and get into the liver. So these are much bigger in mice than they are in people. In people, they're about 80 nanometers in diameter. And so if you want to develop a therapy for the liver in people, you actually need to make sure that you have a nanoparticle size that's going to fit through those holes. Whereas in mice, you could get away with something bigger. So size is important in certain contexts.
So there are already some FDA-approved gene therapies that use viral delivery systems. Is there something better about using these about these lipid nanoparticles than what's already sort of out there?
Sure. Yeah. I mean, there are pros and cons to everything. And some of the advantages of gene therapy is its longevity. So the effects can last, but sometimes they don't last forever and you need to do a repeat therapy. And the problem with viral therapies is that your body will mount an immune response against the virus that's used to do the delivery. And so it's once and done. You better hope it works very well on the first injection, because you're never going to be able to receive a gene therapy with one of those viruses again. The other main limitation of gene therapy is that different proteins and the nucleic acids that go along with them, they're different sizes. And these viruses have a cargo capacity that's much smaller than lipid nanoparticles. And so with lipid nanoparticles, we can fit larger genes if we want to, and we don't have the same risks of immunogenicity as you would with some viral vectors. There are disadvantages, though, too. Viruses can be quite effective at getting to organ targets that lipid nanoparticles have had a hard time with, but hopefully we're here to fix that.
Well, thank you again, Katie, for this very thought-provoking and informative hour you've spent with us. If there are any questions, I do suggest that you get them into the Q&A as quickly as possible. We won't be able to answer any more live today, but we could certainly follow up with any to answer any of the questions you have. I'd also like to thank everybody in the audience for participating today. Great questions. Some of them I wasn't even sure what I was asking, so that's always a good sign. And I hope you'll all be able to join us again for our future faculty insight programs. Please keep an eye out for a follow-up message that'll come in the coming days that'll include a link to this presentation and also some other resources, so you could check out more of Katie's work and get to see what she and her students in the lab are doing. So thank you all for being with us. Again, thank you so much, Katie, and have a wonderful afternoon to everybody. Thanks.